Hello, this is Michael from Calabasas Online Tutoring, and today I'm going to be talking about our first biology video. I'm going to be making this on protein synthesis. Now basically what protein synthesis is it makes proteins in your body which basically make up your entire body. Everything you see on yourself is made up of proteins, so they are extremely important. Now protein synthesis starts in the nucleus. So let's say this is a human cell, and that's the Golgi body, and that's the ER. So in the cell, in the nucleus, there's a section called the nucleolus, and this houses the chromosomes. And these chromosomes, let's make a zoom in section. That's basically what they look like. And basically what a chromosome is made of is tight strands of DNA, which is the code book, surrounding proteins called histones. So this is a histone, and the DNA wraps around it very tightly. So histone. Now this DNA is going to be coding for the protein. Now out here in the ER, well actually before we get to that, there's 3 types of RNA, which is basically a single stranded version of DNA. There's rRNA, tRNA, and mRNA. rRNA builds these things called ribosomes, and they're in the rough ER, which is the section closest to the nucleus. There's this part called the smooth ER out here, which leads to the Golgi body. Now, these ribosomes are very important. They are basically the workbench in which protein synthesis happens. So without them, protein synthesis wouldn't be able to occur. Now, rRNA makes these ribosomes. And then once those are made, that signals to the DNA to start making some mRNA, which stands for messenger RNA. So the DNA, it has the nitrogenous bases over here. Let's say it's AUG because that's the most common, and then that's the most common starter for protein synthesis, and it's gonna keep going, CUA and so on. So basically, messenger RNA binds to this like this, and it has the complementary pair. So in RNA, as you may know already, in DNA, adenine, which is A, binds to thymine, and guanine binds to cytosine. But in RNA, thymine is replaced with uracil. So the U stands for uracil, A stands for adenine, G stands for guanine, C stands for cytosine. Now, A will always bind with uracil, so we're going to put uracil on top, and then uracil always binds with A, and then G right here, that always binds with cytosine. So we have an mRNA with 3 things that start with U, A, and C. Now, that is called, that section of 3 nitrogenous bases is called a codon. So we have our mRNA, UAC, and this is our codon. And there's multiple sets of these codons throughout the entire strand of RNA, of mRNA, until it gets to this thing called a stop codon, which is where the strand ends. Now, we mentioned the ribosomes earlier, and the ribosomes are split into two parts. There's a big part and a small part. The mRNA comes out of the ER. It comes out of the nucleus, and it goes in between the two sections of the ribosome. And so here's the codon right here. We still have uracil adenine cytosine, and it's sitting there waiting for another strand of RNA called tRNA. This stands for transfer RNA. Sorry. And basically, transfer RNA is shaped kind of like a clover. And it has an amino acid on top, usually methionine. So the first tRNA strand usually will have a methionine on top, and at the bottom of the tRNA you see these three things again, but this is called an anticodon. And these are the complementary base pairs of the mRNA. So, as we can see, U is right here, so A will be right here, because A goes with U. And then A is right there, so U goes here. And then G goes here, because G goes with C. Now, we have our mRNA waiting with its codons. And it has more and more until it gets to stop one. In between the ribosome. So, the tRNA will come. Here's the mRNA still. The tRNA will come, and there's three sections in which it can be. There's the E side, the synthesis side, and the A side, which is the acceptance side. So, the first strand of tRNA, it comes down with its amino acid, and it binds to the mRNA, and the mRNA stores that information inside of the ribosome. And then this tRNA waits for another tRNA to come. So we have this codon right here. The ribosome shifts over one codon, which means that this tRNA is now over in the E side, and then another tRNA comes with its anticodon and with another amino acid. Now, once this second tRNA binds, that means this first one can go. So everything leaves except for the amino acid. The amino acid stays behind. So it's floating here. And it's waiting for something to happen. So we have our ribosome over here with mRNA in there. This codon has already been coded for, and this codon is being coded. Now, once the ribosome shifts its one codon again to go, then that'll mean this amino acid also is free. And what happens when two amino acids are free next to each other? They bind with a peptide bond. And so this ribosome keeps traveling down this strand of mRNA until it reaches a stop codon, which doesn't have a tRNA with an anticodon for it. And it just ends up making this long chain of amino acids. And once it reaches the stop codon, once the ribosome reaches it, and this long strand is made, a long strand of amino acids is a protein. So, through that process, that whole long process, that is what gives us our protein. And this happens multiple times until it makes more proteins, and then things can attach and make and make tissues, and make organs, and make organisms, and so on. And so, as you can see, protein synthesis is a very, very important thing while complex. And I hope you understood some of it. If you don't, ask us questions, ask us to elaborate more. But thank you so much for watching. I really hope you learned something.